So far, we've been talking about Mendelian genetics, and so now we want to shift gears a little bit and talk about some variations on Mendelian genetics. And before we dive into those, I just want to, for the sake of comparison, let's just talk about what we've been dealing with to this point. So what we've been dealing with is a phenomenon called complete dominance. Okay, and that's illustrated in this slide, and it's what we're really familiar with when we do basic Mendelian genetics. So again, if you look at this slide here, you see there are two different phenotypes associated with this flower. There's either purple or white. And what you also notice is that the heterozygote is indistinguishable from the homozygous dominant plants. And so this is what we would refer to as complete dominance. There are two different alleles here, capital P and lowercase p. You get two very discrete traits, and these are all what Mendel was looking at. Remember, he looked at tall and short, purple and white, round and wrinkled, so very discrete, either this or this, and there was no kind of in between. And again, in this case with complete dominance, which is what we've been talking about, the homozygote and the heterozygote are, the homozygous dominant and the heterozygote are phenotypically indistinguishable. And in this case, what we now know, in this particular example, is that the gene that we call P encodes for a protein, we're calling P there at the bottom of this slide, and what that protein does is it converts colorless to purple. And so all you need to have is one allele that codes for the protein P in the heterozygote, and you become purple. And so again, this is complete dominance, and this is what we've been talking about up to this point. So now I want to talk about variations on that and some things that we refer to as modifications of dominance. So in this slide, we see one of these modifications of dominance called incomplete dominance. And in the case of incomplete dominance, neither allele, again we're looking at two alleles here, but neither allele is dominant or recessive. And what you have, instead of two different very discrete traits like we saw on the previous slide, there are actually three traits. So two different alleles, and you see them on this slide indicated with with C and then a superscript. Because remember, we use the capital and lowercase letters to deal with dominant and recessive alleles, but in the case of incomplete dominance, neither allele is dominant or recessive, and so that lowercase, uppercase letter thing just doesn't hold, and so we often do it this way, with a letter and then a superscript. So in this case, we have two different alleles, C superscript R and C superscript W, and what you notice is that in the F1 with the heterozygote, the phenotype is not the same as either one of the parents. It's actually in between, and sometimes it's kind of a blend of the two. And you can see that in the F2 at the bottom, and again we're looking at this example of 4 o'clock flowers. In the F2, if we took two F1s that are both heterozygous and we cross them, you would get the Punnett square that's shown at the bottom of this slide. And what you notice then are those that are homozygous for the CR allele would be red. Those that are homozygous for the CW allele are white, but the heterozygotes don't look like either one of those. They're actually somewhere in between. They're an intermediate between the two homozygotes, and this is a classic example of what we would call incomplete dominance. And in this case, your ratio of genotypes, right? You've got 1 to 2 to 1. One homozygote to two heterozygotes to one homozygote is the same as your phenotypic ratio, right? You have 1 red to 2 pink, in this case, to 1 white. And so in the case of incomplete dominance, you see your genotypic ratio and your phenotypic ratio are the same. Another classic example of a modification of dominance, so we just talked about incomplete dominance, is codominance. And the best illustration of this in this example is human blood typing. And we'll talk about that here in a second. But again, we're dealing with, in this case, there is often no dominant or recessive allele. In the case of the red human blood type, there is actually a recessive allele. But what I want you to focus on here are the capital letters I with the superscripts A and B. And what we notice in this case with codominance is that both of the alleles are expressed in the heterozygote. So go down to the middle of this slide and you look at where we have the heterozygote. We have capital A superscript, or capital I, sorry, superscript A and capital I superscript B. That's the genotype of people who have the blood type AB. And codominance really means that both alleles are being expressed. And so one, again, is not dominant over the other, but they're both expressed. And in this case, with the blood group, what we're talking about are different sugars that are expressed on the surface of red blood cells. And so, again, we see different blood types. If one carries the IA allele, they would have A blood type because they're expressing that type of sugar on the surface. It's indicated here with triangles. And if they have the B allele, they would be expressing the B sugar type, and that's shown here with little circles on the red blood cells. And then the codominance is really when you put those two together, and you see both of those sugar types being expressed on the surface. So it's not a blend, it's both of them being expressed. And then you also have a recessive allele in this case, the lowercase I, and that doesn't express any sugars, and that would be the genotype of individuals who have O-type blood. And we'll come back and talk about these different alleles as they relate to human blood type again in a second. What I want to do next is just, again, reiterate the difference between incomplete dominance and codominance in the next slide. So this slide, I think, really nicely illustrates, by looking at flower color, the difference between incomplete dominance and codominance. So again, in the case of incomplete dominance, if we took a red flower and a white flower, the heterozygote, which is shown on the bottom left here, would be pink. And so it's intermediate, it doesn't look like either one, and it's almost a blend of the two alleles. But again, neither the red nor the white is dominant, but you have a different trait, again, expressed in the heterozygote. And when we're talking about codominance, you see that in the bottom right. So in that case, if we crossed a red and a white flower, you would actually see both of the phenotypes, the red and the white phenotype, expressed in the heterozygote. Again, neither one is dominant or recessive, but they're both expressed, and it's not as visibly a blend as you see in the case of incomplete dominance. Let's go back and revisit the human blood type example. Another example of a modification is situations where some alleles actually might be lethal. And by lethal, I mean when certain alleles are in combination, the organism doesn't survive. And a classic example of that is shown in this slide here. I don't know how many of you are cat aficionados, but there's a somewhat famous breed of cat called the Manx cat, and they're tailless. And part of the reason why the reason they're tailless is because there's a gene that influences their spine development, and that gene has an interesting allele. And so cats that are heterozygous for that lowercase t allele that's shown here, you can see they don't have a tail. And so heterozygotes are these tailless Manx cats. So if we took two tailless cats, Manx cats, and mated them, you would see the results that's shown in the Punnett square there. And normally we would expect then, obviously, 1/4 would be homozygous dominant, 1/4 homozygous recessive, and then 2/4 would be the heterozygotes. Well, what you notice when you do that is your ratio of normal cats to tailless cats is a 1-to-2 ratio, as opposed to the expected sort of 1-to-2-to-1 or 3-to-1 ratio, depending on how these phenotypes play out. And that's because the homozygous recessive in this case are never born. So that bottom right square is just completely absent. And so you only see then 1/3 of the cats will be born normal. Those are the homozygous dominant Ts. And then 2/3 would be Manx cats because the fourth category doesn't ever even appear. And so instead of a 1-to-2-to-1 ratio, or maybe a 3-to-1 ratio in some cases, you see a 2-to-1 ratio of a Manx to a normal cat in this case. Before we leave this topic and start talking about more complex traits, I just want to also remind us that there are, of course, environmental influences to what we see in phenotypes. It's not always, it's certainly not always completely related to the alleles or the genotypes that we see. For example, if you take a bunch of plants and they have exactly the same genotypes, but you grow some at lower temperatures and others at higher temperatures, depending on the plant you use or the plant you're using, you can see varied effects in growth rate, as is shown in this graph here. So this plant seems to thrive at about 75 degrees Fahrenheit, and you see lower or shorter plants at high temperatures, sort of medium-sized plants at the 45 degrees. And so certainly there are environmental influences, and temperature, as is shown in this slide, is one of them. Certainly diet, when we talk about height in humans too, diet can have a big effect on the height of humans. When we look at people who were raised and grew up in countries where nutrition and caloric intake was limited due to limited food sources, you see in general people are shorter. Whereas in countries where there are fairly high protein diets, in general, people tend to grow taller, regardless of the genotype. So environmental influences certainly have an effect as well. So I want to talk about a couple of specific examples of that, in addition to that really show, again, another one that shows the effect of diet. So in this slide, on the far left, one other really great example, I like to show this one because it really illustrates a number of things. Another example of environmental influence on a particular phenotype or trait are what we call sex-influenced traits. These are not sex-linked traits. I'm not talking about sex linkage. These are traits that are autosomal. The genes that influence these traits are not on the sex chromosomes. And one of the most common ones, that's a great example, is male pattern baldness. You can see the top of the head of this male here. And sex-influenced traits, in this case, the phenotype is actually influenced by hormones. Of course, males have much higher levels of testosterone than females do, and because of those different hormone levels, the phenotypes are different in men and women, and men tend to show this male pattern baldness more than women do. So what we're looking at here, we have two alleles, the B plus allele and the B minus allele. And if we just focus on the male side of this table, what you see is men who are homozygous for the B plus allele tend to show this baldness pattern, and men who are heterozygous for that B plus allele also show that baldness pattern. So that clearly is indicating that the B plus allele shows dominant inheritance in males. So remember, we always look at the heterozygote, and in this case, the heterozygote is similar in its expression to the homozygous for B plus. Therefore, B plus must be the dominant allele in males. And when you look at the homozygote for B minus, you see a different phenotype. Right? So this is clearly showing dominant. B plus is dominant in males. So in the next slide, then, let's look a little more carefully and closely at what we see in females in this case. If we focus then on the female side of the story, so same genotypes, right? A female who's homozygous for B plus, heterozygous, and then a female who's homozygous for the B minus allele. What you see is the B plus allele actually shows what we would expect of the recessive allele in females. So again, we always look at the heterozygote to tell us what is dominant and what is recessive. And when we look in females, the heterozygote, the B plus B minus, is similar, the phenotype is the same, really, as the B minus homozygote. So if we look at females, what that is showing is that the B minus allele is showing dominance. So the B plus allele is recessive in females, but the B plus allele is dominant in males. So this is a really interesting sex influence trait. And again, testosterone levels are higher in males, and so the effect of this particular B plus allele tends to have a greater phenotypic effect and show dominance in males than it does in females, who have obviously a lower testosterone level. So those are a couple of interesting examples of how environmental influences also, whether that be diet or temperature or whatever, or certainly hormone level, can also influence what we see in phenotypes. Okay, so now I want to switch gears a little bit and talk about what we call complex traits. And to this point, we have been really focusing primarily on sort of more what we would call simple inheritance. And this is where single genes control single traits. And so let's just focus first on the panel there on the right where it says no epistasis. You've seen this before. This is when we were talking about dihybrid crosses. And yes, we're following two genes here, but each one of the genes controls a single trait. So the gene we're referring to as Y here is controlling the color of the seeds. It's either yellow or green. And the gene we're referring to here as R is controlling sort of that shape of the seed, the pea. It's either round or wrinkled. And so, yes, two genes are being followed at once, but both of them independently affect or control a single trait. And actually, when we think about this, especially in humans, this single gene affecting a single trait is actually quite rare. What we often see, or more commonly see, especially in humans and more complex organisms, is that there are often traits that are controlled by two or more genes. So in other words, genes don't work in an organism in a vacuum, right? What the product of one gene has an effect on the product of another gene, and they work together in concert to affect a trait. So traits are often controlled by two or more genes. And so I want to spend some time talking about that, and that's kind of what we think about when we talk about complex traits. And so the example that's shown here that we're going to look at first is a phenomenon called epistasis. And the panel on the right, on the left, is showing you epistasis occurring in a type of flower called the sweet pea flower, and when we look at flower color, white or purple. And the term epistasis, if you break it down, the prefix epi is on top of, and stasis kind of means to stand. And so epistasis is a phenomenon where what's happening at one gene masks or hides or somehow impacts the expression of the alleles at a second gene. And so that's what we're talking about when we talk about epistasis. So let's look at this example diagrammed here. So we have two varieties of white flowers. We're calling them variety 1 and 2. And we can see that they're both true breeding when you look at the genotypes. Yes, variety 1 is homozygous dominant at C and homozygous recessive at P, but that's still true breeding, right? If I took white variety 1 and I continued to self-cross it, all I would ever get back is white variety 1 with that same genotype, homozygous dominant at C and homozygous recessive at P. And the same is true for white variety number 2. So they're both true breeding parents. And then what kind of alerts you to something different going on here is when you cross these two true breeding white flowers in the F1, or plants with white flowers, in the F1 you get a purple colored flower, which is a little bit of a head scratcher right there. And then when you self-fertilize that F1, you get the results that are shown at the bottom panel there in that Punnett square. And if you count up the number of purple flowers and the number of white flowers that arise in the F2s, what you get is this sort of strange 9-to-7 ratio. And so if we compare that to what we would expect in the case of no epistasis or no gene interaction, where we would expect the 9-to-3 to 3-to-1, 9-to-7 could be, thought of as a variation on that. But when we do this kind of cross with sweet pea flowers, you don't get that 9-to-3 to 3-to-1, and you don't get a 3-to-1 either that you might expect with a monohybrid cross. You get this 9-to-7 ratio. So what's happening here? So if we dig into that Punnett square a little bit deeper, And I've drawn this out for you in the class notes. But if we look at what do all the purple squares have in common? Well, all the purple squares in that bottom panel there have at least one dominant allele at C and at least one dominant allele at P. So dominant at both C and P results in purple. And everything else results in white. So again, if we go back and think about what were the genotypes that gave us those 9 to 3 to 3 to 1 ratios, if we had a dominant allele at C, but were homozygous recessive at P, we expected that genotype to appear in a 3/16 ratio. And in this epistasis example, those are all white. Those appear white. And the same is true if we're homozygous recessive at C and then dominant at P. We expected a 3/16 ratio of that genotype, and in this example, those are also all white. And then the same is true for the organisms that are homozygous recessive at both genes, where we had an expectation of 1/16 of those organisms should have that genotype. And again, those are all white. And so if we add those three categories of the two 3/16 categories and the 1/16 category, that's where we get our 7. And so all the organisms that have genotypes that are not at least one dominant allele at C and one dominant allele at P result in white. So we get this 9 to 7 ratio. So we must have a dominant allele at both of those genes in order to get the purple pigment. So the next sort of interesting thing to ask is, what's going on here? Why is this happening? And so let's talk about that and look at that more specifically in the next slide. Okay, so on the far left, we see the self-fertilization. So that's the F1, right? We self-fertilized the two purples, and then the arrows there are pointing to the expected, 9 to 3 to 3 to 1 ratios of different genotypes, right? What is the ratio that's going to have dominant at both C and P? That's 9/16. What's the ratio that's going to have dominant at one, recessive at the other, 3/16, recessive at one, and dominant at the other, okay, 3/16, and then recessive at both is 1/16. So we talked about that in the previous slide. So what if the production of anthocyanin, which is the purple pigment, okay, I don't care that you remember the name anthocyanin, it's a purple pigment. What if that is a linear pathway that's shown here? And so there's a precursor or some sort of molecule that needs to be converted. Precursor 1 needs to be converted to precursor 2, and then precursor 2 gets converted to anthocyanin or purple pigment. And there are enzymes that catalyze those steps. Okay, so what if there's an enzyme, right? We know enzymes are proteins. An enzyme that we're going to call enzyme C that's encoded by the gene we're calling C. Well, in the top panel there, shown in blue, you have the arrows. If we have a dominant allele at C, we can convert precursor 1 to precursor 2. And if we have a dominant allele at P, that enzyme is working, it's functional, so the dominant alleles represent functional enzymes, then we can convert precursor 2 to anthocyanin or purple. And as long as we have functional C and functional P, we can work our way through that pathway and end up with purple-colored flowers. But now what if we have homozygous recessive alleles at one of the two genes or at both? That's what's shown in the next three rows there. So recessive alleles, let's say that they produce non-functional enzymes. So in other words, there where they're red and they don't show the arrow, they show that line across. That means that the enzyme is not working and it cannot catalyze that conversion. So if we have a dominant allele at C, yes, we can convert precursor 1 to precursor 2, but if we don't have functional P, there's no way to get purple pigment. And so that would result in white. And then the converse of that is in the next row where we can't convert precursor 1 to 2, right? If we can't do that, then we certainly still can't get purple pigment. And at the very bottom, if we don't have either one of the enzymes fully functional, then we certainly cannot produce purple pigment. So this is sort of an explanation of why we see the inheritance patterns that we do, and it's directly related to what's happening at the molecular level. So as long as we have functional C and functional P encoded by the dominant alleles, then we can get purple. Anything other than that blocks this pathway at one step or the other and doesn't allow purple pigment to be produced. And if precursors 1 and 2 are colorless or white, then we would end up with this 9-to-7 ratio. So that's a molecular explanation of the genetics that we saw on the previous slide. So let's look at another interesting example of epistasis. So let's look at another example of epistasis. And this one is a pretty classic example. Some of you may have heard of this one before and may be familiar with it a little bit. But this is how epistasis works in the color of the coats of Labrador Retriever dogs. And again, what we're looking at here is an example of what we would expect, and we have two genes working here. So let's say we take two black labs, and black labs have the genotype shown there. They're heterozygous, have both genes that we're referring to here as B and E. And if we mate those two black labs, we get this strange ratio that's shown here in this Punnett square. We don't get a 9-to-3 to 3-to-1 that we might expect if there was no epistasis. We don't get a 9-to-7 like we talked about in the previous slide, but we still get some kind of strange variation of that 9-to-3 to 3-to-1, and instead we get a 9-to-3-to-4. So if we count up again the squares here, 9/16 are black colored dogs, 3/16 are brown colored, and then 4/16 would be yellow labs. And if we, like we did before, dig a little deeper into what the genotypes are that are represented in each one of these squares, and I've written this out for you in the notes, what we notice is if these dogs have a dominant allele at both B and E, then they will be black labs. Okay, so black is dominant at both, and that's our 9/16 portion of this. And then what's the genotypes of those that are brown or chocolate labs? Well, what we notice when we look at that group is that those are all the dogs that have homozygous recessive at B, but they do carry at least one dominant allele at E. But they're homozygous recessive at B. And we would expect that to be the 3/16 ratio or category. And then what are the yellow labs? What are the genotypes that are the yellow labs? Well, if you look carefully, they're all the genotypes that are homozygous recessive at E. And it doesn't really matter what they are at B. I mean, you could be homozygous dominant at B, heterozygous at B, or homozygous recessive. And as long as you're homozygous recessive at E, you would be a yellow lab. And so that group composes both a 3/16 category and the 1/16 category. So there's our 4/16. This is our 9-to-3-to-4 ratio. So if we think about this, I mean, this is what I find really fun about genetics. All this inheritance pattern, that's all fine. But what I find really interesting, and I hope some of you will too, and go on and continue to learn more about this, is why. The question of why. What is happening at sort of the molecular level that gives you this interesting inheritance pattern? And what we do, I'm oversimplifying this a little bit, but just to sort of explain what's going on. What we know about the B gene is the B gene is really involved in the production of pigment, right? The black or the brown pigment. And what we notice is if you have a dominant allele at B, you produce more pigment, right? This is the black labs. Whereas if you have homozygous recessive at B, you get that chocolate color. So the dominant allele just produces a darker pigment, and the homozygous recessive just produces that lighter pigment. So B is involved in the production of pigment. But what's going on in E that we get this strange thing when E is homozygous recessive that no matter what at B, we always get yellow? Well, E is important for the deposit of pigment in the hair of the dogs. And if we say that the recessive allele in this case is non-functional, so if the gene that's important for getting the pigment to the hair is not working, that protein is just absent, then it doesn't matter what pigment is actually being produced, right? So if I can't deposit the pigment in the hair, it doesn't matter whether the pigment being produced is black pigment or brown pigment. It's not going to get into the hair, and so this is why we see the yellow dogs. So B, the B gene is important for the production of pigment. Dominant alleles give you darker pigment than recessive alleles. And the E gene is important for getting that pigment into the hair. And if you have no dominant functional alleles at E, you can't get the pigment into the hair, and the result is a yellow dogs. So again, a really classic, interesting example of epistasis, and then there's always these interesting molecular explanations that go with it. So I'm just going to summarize our two epistasis examples that we just went through. So we talked about the sweet pea color and the Labrador Retriever color, giving us these different variations on that 9:3 to 3:1 ratio. Yeah, and so in this slide, I'm just summarizing everything we just talked about. So for the sweet pea flower color at the top here, in order to have purple pigment produced, you need a dominant functional enzyme at C, and you need a dominant functional enzyme at P. And anything else does not allow you to make your way through that molecular pathway to the end, which is purple pigment. And so all the other options where you lack functional P, or you lack functional C, or you lack both of them, are going to always result in white color. And so you've got two genes at work here, but both of those genes are influencing a single trait in this case, which is the color of the flowers in these sweet peas. And at the bottom, I've summarized what we just went through with the Labrador Retrievers. So in this case, it's a little bit different than what we talked about in the sweet pea flower. But if you have dominant at both B and E, you get the black labs because you're producing the dark pigment at B, and that dark pigment can get into the hair at E. And the chocolate labs, you still have a functional E, but you have less pigment being produced when you're homozygous recessive at B. And then with the yellow labs, it doesn't matter what's happening at B because you can't get the pigment into the hair if you're homozygous recessive at E, so you see the yellow labs in all those cases. So we've got, the 9:7 ratio at the top and the 9:3:4 ratio at the bottom, and these are all variations on a dihybrid cross where instead of a single gene influencing a single trait, you have two genes in this case, in both of these cases, influencing a single trait. And these are classic examples of epistasis. Okay, so we've talked now about how we can look at two genes and how they interact to influence a single trait. So I want to just talk briefly about this general category that's also under this variations on Mendel called polygenic inheritance. So poly meaning many. So what about the situations where there are more than two genes that influence a single trait? What if there are many genes that influence a particular trait? And this is actually the case with most traits, certainly in humans. If you recall when we first started talking about Mendelian genetics, remember when he chose the traits that he was going to follow, right? The purple and white or the round and wrinkled. He chose very discrete traits. And even when he chose tall and dwarf, he didn't have, tall, kind of tall, medium tall, right? Short, kind of short. He chose he didn't have, white and light purple and dark purple. He chose very discrete traits that were either this or this. And that is not, again, the typical situation with most traits, especially in humans. And so many traits show continuous variation across a whole range. In other words, they're not discrete. And human height shown here is a classic example. I mean, you don't look at somebody and say they're either tall or short, right? There's everything in between. You have tall people, you have what we might consider short based on an average, but then there's this whole continuous range and variation. And these polygenic inheritance traits are single traits that are controlled by many genes. I mean, I don't know how many genes are involved in controlling human height, but it's certainly more than two. So there may be, different alleles at 5, 6, 7 different genes that are involved in human height. And when you think about in a population, all the possible combinations of those different alleles at 5 or 6 different genes, you can come up with sort of this normal distribution of height. And so the phenotypes that we see when you're talking about polygenic inheritance, sort of a combined or cumulative effect of all kinds of different alleles at many, many different genes. And human height is one example of that. But the other thing that you also have to consider when we're talking about this particular trait is certainly nutrition. We talked about that earlier. We know that individuals who were raised under conditions of poorer nutrition tend to be shorter. We also know, and you can see in this inset here, that men tend to be taller than women. And so sex hormones also influence. So you've got polygenic inheritance, lots of different genes, lots of different alleles affecting a single trait. And on top of that, you have environmental effects as well. And in this case, nutrition and sex hormones. And so then there's one other example I want to talk of polygenic inheritance that I want to talk about in the next slide. So this is another example in humans, polygenic inheritance, many genes influencing the trait, in this case, of skin pigmentation. So what we're looking at here again is a normal distribution, and I think in this particular case, it's indicating that there are 3 genes involved in skin pigmentation, indicated here by A, B, and C, and there are different alleles, some that have a superscript L indicating light, superscript D indicating dark. And so you can imagine how many different combinations. So we're just looking at 2 different alleles at 3 genes here, right? And so you can see all the possible combinations, and the more dark alleles you have, the darker your skin pigmentation. If you look on the far right there of that normal distribution, you see A, B, and C are all homozygous for the dark or the D alleles. And if you look on the far left, you see all A, B, and C are homozygous for the L or the light alleles. And then there's all these possible combinations in between. And so this is what kind of gives you that normal distribution. So again, we don't look at people and say, oh, they are really light pigmented and everyone else is dark pigmented. There's this whole continuum, this normal distribution of ranges of the color of people's skin. And just as we talked about with the height in the previous slide, environmental influences also have an effect on skin pigmentation. We know that populations in sunnier environments shift this entire curve a little bit more to the right with darker skin pigmentation. So, and again, I don't know if it's just 3 genes that influence skin pigmentation. What if it's 4, 5, 6, and there are more than 2 alleles at each one? You can just imagine how many different possibilities there are, and it results in sort of this continuous variation over a range. And these are this is very, very common. It's less rare to have the discrete traits that Mendel worked with when he was working with the P's. Polygenic inheritance certainly is a much more common phenomenon. Okay, one last variation on Mendelian genetics that I want to talk about, and we're just going to touch the surface of this one and not get into too much detail. But this has to do with what we call genetic linkage. And so I think it's not a stretch to know and understand that there's more than one gene on a single chromosome. So, I mean, you think about it, in humans, we have, 23 different chromosomes. We have 46 in a diploid, but there are 23 different chromosomes. And we know we've got, 25 to 30,000 different genes. So clearly, in order for that math to work, there are more than one gene on every chromosome. So how does that affect inheritance patterns? And so this is just, in this slide here, it's a picture of a chromosome from flies. And I don't know which chromosome number this is, but you can see there's a bunch of different genes on here. They're showing some wild type and some mutant phenotypes that are associated with them, but you can see there's a gene on this chromosome that determines body color. You can see there's some gene on here that has something to do with the wing shape. A couple of them that have to do with wing shape. Vestigial wings is on this one. You can see some other eye color ones. So clearly there's more than one gene on a single chromosome. So how does that affect what we see in inheritance patterns when we're looking at the inheritance of linked genes? And Mendel kind of got lucky because I don't know if he intentionally chose the genes he was looking at this way, but we now know that basically all of the genes that Mendel was looking at when he was doing his initial experiments were not linked. They were on different chromosomes, basically, so that's why his results came out nice and clean. But we know that's not the case always. There are linked genes. So what does that mean, and how does that affect meiosis? Let's look at that in the next slide. So in this slide on the left, we're looking at what we've been looking at so far over and over and over again, right? Independent assortment, unlinked genes. So in this case, we have two genes, A and B, and this diagram on the left here is showing an individual who's heterozygous at both of those. So they carry the dominant allele A on one homolog, which is the dark blue, and the recessive allele A on the other homolog, which is shown in a lighter blue. And then you can see they're heterozygous at B as well. But A and B are on different chromosomes. And so they assort independently. And we've talked about this quite a bit. So the homolog with the capital A could segregate with the dominant B, but it also could segregate during meiosis with the recessive B. So there's all those possible gamete combinations that are listed there. And that's what we've been talking about so far, independent assortment with unlinked genes. So what happens when you look at the right side? What if A and B are on the same chromosome? So if you look at this individual here, now we're just looking at two chromosomes, and they're homologs of one another. This individual has the same genotype as the one on the left, right? They're heterozygous at A, and they're heterozygous at B. But A and B are on the same chromosome. So what that means is that A and B are going to go together much more frequently. So if the dominant alleles A and B are on one chromosome, they are going to tend to go to the same gamete more frequently than they would if they were on different chromosomes. And the same is true for the recessive alleles A and B. Because they are on the same piece of DNA, they will tend to go to the same gamete more frequently. And so basically, when we're talking about linked genes, or genes that are on the same chromosome, they don't assort independently. They tend to go together. The alleles tend to go together more frequently than you would expect if they were assorting independently. And you can see what kind of effect that has on the genotypes of the gametes in that panel on the right there. So let's look at some real examples, I think, to really illustrate how we can detect and look at inheritance patterns and determine whether genes are linked or not. Let's look at some experiments that were done and ask a few questions and see if we can't work through this a little bit. So in this slide, we have one of the first demonstrations of genetic linkage. And so again, we're looking at two traits here. We're not compounding this or confounding it with epistasis or any of the polygenic stuff we've been talking about. We're just looking at, again, now two genes, and each one influences a single trait. And in this case, we're looking at flower color. They're either purple or red. And we're looking at pollen grain length. They're either long or round pollen grains. Okay, and so purple is the dominant phenotype, and you can see that because when we cross the purple and the red, we see all purple flowers in the F1. And we can also see that the long pollen is the dominant trait because, again, we see long pollen grains in the F1s when we crossed the two true breeding parents in the P0. All right, so now let's take our F1s, like we've done many, many times before, and if we self-fertilize those, and we come up with the F2s, what I want you to think about is what, given everything we've been talking about so far, we've got two true breeding parents, there's no epistasis going on here, nothing like that, and we cross them and we get F1s, and then we self-fertilize those heterozygous at both genes, those F1s. What normally would we expect to see? What kind of ratio would we expect to see? And then we can also then, if we had, let's just say we had a given number, there were 427 offspring in the F2s, how many of each phenotype would we expect to see? So before I go on to the next slide, think about that for a minute. What is the expected ratio here? If we're talking about independent assortment, two different genes affecting the two different traits, and we cross or self-fertilized the two F1s. And then once we think about that ratio, if we have 427 offspring, how many offspring should we expect in each phenotypic class? So take a minute and think about that before we move on to the next slide. Okay, so on this slide in red, I've kind of worked all of that out. So if everything was going as we talked about before, and this was a typical dihybrid cross, we would expect that 9:3 to 3:1 ratio that we've been talking about many times before. And going back to, again, what we've talked about many times before, we would expect 9/16 to have both of the dominant phenotypes, which in this case is the long, or the purple flowers and the long pollen. And we'd expect 3/16 to have purple short and 3/16 to have red long, right? One recessive, one dominant. And then we'd expect 1/16 to have both of the recessive phenotypes, the red flowers and the short pollen. And we can take our 427 offspring and figure out what fraction of that 427 would we expect to see in each of those phenotypic classes. So you take your expected fraction and you multiply it by 427. And you guys did something similar to this when you were doing the chi-square lab, the analysis of the fly data, when you had a model or a prediction or an expected ratio, and given a certain number of organisms, how many organisms then would you expect to see in each class? And here are the numbers. We would expect 240 that have both dominant traits, and 80 that have one or the other of the dominant and the recessive. And then we would expect 27, 1/16 of 427. We expect 27 would carry both recessive alleles. So this is what we expect if independent assortment was going on. So this is our hypothesis, right? We're expecting a 9:3 to 3:1. If we had 427 offspring, we should see this kind of distribution. So we do the experiment, and then we look at whether or not the data actually support this hypothesis of 9:3 to 3:1, or unlinked genes in this case, right? Genes on separate chromosomes. This is what we would expect. So do the data that are actually collected support this hypothesis of independent assortment, or genes that are not linked, or in other words, genes on different chromosomes? Well, let's look at the data that was collected and see if we can answer that question. So in this slide, I've highlighted what was actually observed in the 427 offspring that arose from this self-fertilization of the F1. And the expected numbers are, I'm looking way at the bottom here, the data that was actually collected. The expected numbers are shown there from the previous slide, right? We expected the 9:3 to 3:1, and the expected numbers, if it was 427 total, are shown there in the expected number column. Well, now let's look at the observed numbers. Well, we had some pretty funky ratios. What is it? 15.6 to 1 to 1.4 to 4.5. And you can see that the numbers do not fit with what we expected. Okay, and these look like they're pretty off. And so what we would say here is, by looking at our observed and our expected, our data doesn't seem to support that hypothesis that these two genes, the one for flower color and the one for pollen length, that these two genes are assorting independently, or that they're on different chromosomes. So this kind of result might predict or support the hypothesis that these two genes are on the same chromosome, because we see something quite different than what we expected as far as numbers go. And so let's look at one other example of this where it's a little bit easier to see. the 9 to 3 to 3 to 1 ratio is kind of, it makes it a little difficult to see how much and whether or not you've deviated from expected. So let's look at one other example that's a little easier to see these kinds of differences. All right, so we're going to do something very similar here, but we're setting this cross up a little bit differently, like I said, to make it a little bit easier to see what, if there are any of those differences that we observed in that previous example. So in this case, we're looking at fruit flies. And the fruit flies here, we're looking at two different traits, same kind of thing, two traits controlled by two different genes. One of the traits is body color, and the other one is wing shape. So the dominant phenotypes are gray body and straight wings, and we see that with the female fly up there at the top in the P0 cross. And then the recessive traits are black body and curved wings, and we see that in the male. These are autosomal. We're not talking about any sex linkage, no epistasis, nothing here. So the male carries is true breeding for those recessive traits. So what we're going to do in this case, instead of taking the F1 and either doing a self-cross like we would do with plants, like we did in the previous one, or if flies, right, instead of taking the F1 and crossing it with a sibling, we're going to perform a test cross here. So think way back to when we were talking about Mendelian genetics, and I defined what a test cross was. And a test cross is always involves one of the parents is homozygous recessive. So what we're going to do in this case, instead of crossing two heterozygotes, we're going to take our F1, which is heterozygous at both genes, and we're going to instead perform a test cross. So we're going to cross that F1 by a fly that is homozygous recessive at both genes. So this is a little bit different. And so the ratio that we expect in our F2 is not going to be the same. And so what I want you to do before we move on is, do the product rule or draw a Punnett square or whatever it is you need to do. And I want you to work out what the expected ratio is here when we do a test cross. It's not 9 to 3 to 3 to 1 in this case, right? Because we're not crossing two heterozygotes. We're crossing a heterozygote at both genes with a homozygous recessive. So the expected ratio is different than 9 to 3 to 3 to 1. So take a minute, think about that. What's our expected ratio here? And given 1,000 offspring, in this case, what are the expected number of flies that would be in each phenotypic class? Okay, so unlinked genes, independent assortment, in a test cross like this, what is the ratio that we would expect? And given 1,000 offspring, how many of each class would we expect to see? So take a minute, see if you can't come up with those ratios and numbers. Okay, again, in this slide, we've worked that all out. So what we would expect to see if we crossed a heterozygous at B and C with a fly that's homozygous recessive at both B and C, we would expect to see 1/4 of the flies having both dominant traits, 1/4 having one dominant and one recessive, and 1/4 of the flies carrying or having both of the recessive traits. So we would expect a 1 to 1 to 1 to 1 ratio. So equal numbers of all 4 different phenotypic classes. Again, this is not the same cross that we looked at in the previous example with the flowers. We our cross to establish or to generate these F2 was different, and so we have a different expected ratio. So make sure you're clear on that when we set up the cross that's shown here, we don't expect that 9 to 3 to 3 to 1. We have a different expectation. And so if that's the case, it's really easy to look at then how many of each phenotype would we expect given 1,000 offspring? Well, if they're all supposed to be there in equal numbers, 1,000 divided by 4, or 1/4 of 1,000, would be 250. So we would expect, if this is the case, If the gene for body color and the gene for wing shape, B and C in this case, are on different chromosomes and they assort independently, if I set up this cross, this test cross, I should see about 250 flies. If I had 1,000 of them, about 250 flies in each of these phenotypic classes. So this is what I would expect if there was independent assortment and these two genes were on different chromosomes. So now let's look at, again, what data we actually collected by doing these crosses. Right, so if we look at the data that was collected here on the bottom of this slide, the 4 different phenotypic classes are listed there, and we would expect an equal number of each of them. And what we actually got, the observed, were quite different from that. So we noticed that we had over 300 of the gray body straight wing flies, 371, and we had 359 of the black body curved wings, etc. So the actual data do not support very well. These seem pretty off. They don't support very well our hypothesis of a expected 1 to 1 to 1 to 1 ratio of each phenotype being present in about equal numbers. And if we look carefully at this, the other thing that is important to notice is which of the phenotypic classes seem to be overrepresented and which of the phenotypic classes seem to be underrepresented. Well, if we look at the ones that are over 250, it's the gray body straight wings and it's the black body curved wings. Those are the same phenotypes as what we observed up in the parentals, right? If we go way up to the top panel, what was the phenotype of those initial parental flies? Well, it was a gray body straight winged fly and a black body curved winged fly. So those parental phenotypes seem to be overrepresented, and then the other 2 phenotypes seem to be underrepresented. And so that's an important thing to think about when we think about what's actually happening here and how this relates to what's happening in meiosis. So we didn't talk about this a lot, but during meiosis, remember homologs come together and they pair up or line up next to each other during metaphase 1 of meiosis 1. And that is where there's an opportunity for crossing over to occur between homologous chromosomes. So if we take a look at this specific diagram, and it relates back to the previous slide. So in the female, right, remember she's heterozygous and she has the wild type plus alleles on her red chromosome and the recessive B and C alleles on her blue chromosome. Those 2 homologs will come together during meiosis, and most often they will separate and you will get the chromosomes in her gametes that are shown, the 2 on the left. When you look at those 4 gametes that are shown there, the red chromosome with the B plus C plus, and then the blue chromosome with the BC. Right? So those are what's going to happen most frequently. And those are what we would call the parental combinations, right? They're the same combinations that were in the F1 female and also in the parentals. But in the rare instance where crossing over or genetic exchange occurs between these 2 homologs, you would get the chromosomes that are shown on the right side of those 4 gametes, right? The ones that have both a mixture of the red and the blue chromosomes. If there was a crossover event and some genetic exchange during meiosis in that female, you would get these recombinant, it's sometimes referred to, or these non-parental combinations, right? The female did not have the dominant B plus and recessive C. She had B plus and C plus. So those are non-parental combinations, new combinations or recombinations of the chromosomes, and those arise by crossing over. So keep this in mind, and then let's summarize this on the last slide here and look at how this kind of all makes sense with what is overrepresented in our data and what was underrepresented in the data that we collected. So this is the same slide that I showed you before. But we're really now illustrating, okay, so we had the gametes from the previous slide, and now we're adding into those on the bottom panel. I'm looking at the F2 again. Notice that every one of those gametes from the previous slide now has been fertilized by the sperm from the male. Those are those purple chromosomes, right? So that's the male that fertilized those eggs. And if you look at the first two that were overrepresented, or those that we're calling the parental combinations, those are the ones that are going to be present in, or are present in a much higher number than expected, because no crossing over established or generated those chromosomes. The B and C genes are on the same chromosome, so they tended to segregate together more frequently because that's the combination that they were in the parental. So those are the parental original combinations. So the only way we get the other two phenotypic classes, those that were underrepresented, is if there was a crossing over event during meiosis in the female. And that's where you get those combinations of red and blue chromosomes shown there. Those are the same chromosomes that you saw on the previous slide. And then when fertilized by the male with those purple chromosomes, those are our non-parental combinations. So if two genes are linked, what you expect, you don't get what you expect to see if it's independent assortment, right? You get that deviation from your expected ratio. But the other thing that is a telltale sign that two genes are linked is that the parental combinations are overrepresented and the non-parental combinations are underrepresented. Because the only way you get those non-parental combinations is if a crossing over event occurred. And that's going to be more rare than if no crossing over occurs. So crossing over happens, but it doesn't happen all the time. And so these are some telltale signs of linked genes, is that, again, they don't fit with your predicted ratios. And in addition, the parental combinations are overrepresented and the non-parentals are underrepresented. And in this fly example, doing a test cross like we did in this one, where you have an expectation of equal numbers in each class, it's much, much easier to see those deviations than it is in that very first example we looked at with the expected 9:3 to 3:1 ratio. It's just harder to see those differences when you have those kinds of ratios as opposed to an equal numbers of expected flies.